Speaking of dimensionality reduction, it's time to do that, and we can do that by different approaches. And here we will look at PCA as linear approach, t-SNE and UMAP as nonlinear. So to do this, first we calculate the principal components, and then we plot the first PCs, which you see here by this function in the scMPy, and then we visualize component 1 versus 2 and 7 versus 8. So these are the distribution, the expression profile in reduced dimension using principal component. We can also check which genes contributing most in each of these principal components. And so one thing which you note is that not all the PCs can capture large fraction, and it's important to pay attention to the values which are above 0. And you can see certain genes have higher contribution to each of these principal components. In order to see which of these principal components, how many of the principal components contribute most to the variation we see in the data, we can use the elbow pattern. So what we do is that we rank, and the ranking looks like this, which shows the variance ratio. So usually with the elbow pattern you see like a break in the ranking. Here is not very obvious elbow pattern. I have written 21, but I think maybe we can go up to 30. So which, based on that number, we can then use it to run t-SNE and UMAP. And t-SNE, now that I just mentioned about t-SNE, is as simple as PCA. You just specify the function, the object, and then the number of principal components. And the next step, you visualize it. Again, you can color it by given variable. So here we are going to use sample. And at the end, we will compare all of this. Let's run t-SNE, and then we visualize it after. There we go, we have the t-SNE plots. As you can see, they are the samples or cells belonging to different samples have been distributed in the space. And you might see that we have some samples that they are scattered around. So the same sample is, for example, these two overlapping with none of the other samples. And it may represent some technical differences. And when we do the data integration, we will overcome this pattern. UMAP is also another approach. So to run UMAP, we need to calculate, these are graph based. So we need to create a distance matrix. So here we use neighborhood, and here is the number of PCs, and the distance matrix is calculated based on the given number of neighbors for each cell, and here we have set it to 30. We run the neighbors first, and then UMAP, and at the end the visualization. So while it is running, I can again, yeah, we have it now. So here we can also see that there is potentially a batch effect that we see in the samples, in the distribution of their cells within this space. So instead of 2D, I mean 2 components on UMAP, we can also increase the number of components when we are calculating UMAP. And in order to keep the original 2D version, we save it into a new variable, which is called UMAP-done, and then we do the visualization and comparison, which I have already run it here. So remember that by increasing the number of UMAP components does not always lead into a clear or more informative view, which you can see that in this picture as well. So here we have only 2 components. Here we have when we are visualizing or using more than 2 components. This is UMAP 1 versus 2, and this one is UMAP 3 versus UMAP 4. So while higher dimensional embedding can preserve more of the global structure, but the added complexity can also introduce a bit noise, which you can see that here as well. It can become relevant for machine learning purposes, but not necessarily for visualization. So we go with the default number of components, which is 2. And last but not least is that, as I showed you, we first calculate the neighbors, or how connected or how similar the neighbors are. And this picture here, with the arrows or with the lines or edges, shows how close or how similar given cells are. The thicker the line, the stronger correlation or similarity exists between the cells. So now let's compare all of these reduced dimensionality approaches that we have generated so far. Here we have PCA and TSNE and then UMAP. So probably one of the main conclusions is the presence of the batch effect, and can have a strong impact on downstream analysis, differential expression. So I would say we can also, at this stage, we can also visualize some QC metrics on UMAP. Just as an example, for instance, here we are showing the total count and also mitochondrial. So it's overall well distributed, but when we look at the percentage of mitochondrial, we can see that there is a cloud here which shows lower. And guess what? It's the same sample that we had seen in the violin plot, showing shifted towards lower mitochondrial RNA or genes. So again, I think we should go forward and then do the data integration too, and we will check how is the distribution at that stage. Just for fun, you can also visualize some genes of interest. You do exactly the same, but here you pass the gene names. And here you see the overall expression of given genes across the cells. So some cells show a stronger expression of these genes compared to others. Let's save the object and then go to the data integration.